let's continue building concepts related with the MHC molecules. We have already discussed in detail about the MHC class 1 molecules, and what we have discussed up to now, let's review it briefly. That MHC class 1 molecules consist of a large alpha chain. Every molecule, MHC class 1 molecule, consists of a, yes, okay, I will draw one, alpha 1, alpha 2, alpha 3. MHC class 1 molecules, which are expressed on the surface of all nucleated cells, they are having one large alpha chain which is coded in MHC region. Is that right? And this alpha chain has alpha 1, alpha 2 and alpha 3 domain, right? And with that there is beta 2 microglobin molecule which is not coded in MHC region. This molecule is genetically coded in chromosome number 15. Is that right? Now, about this molecule, that this molecule has a pocket for binding antigenic peptide, right? Special type of peptide fragments can bind over here, right? And these regions vary from molecule to molecule, right? Then they have one more region on the molecule which determine the allelic specificity, right? This molecule, this part of the molecule is different in type 1A, in B and C, and also they are different for the different alleles present within the A molecule. For example, A1, A2, A3 molecule will have slightly differences here. And this area is the constant area in all the class 1. All the class 1, either they are A or B or C molecule, or they are maternal or paternal, they are in any human being, right? This part of the class 1 molecules, you can say this is universal part of the class 1 molecule or constant part of the molecule. This specially functions as a identification point or interaction point with CD8 molecules in T cytotoxic lymphocytes. After that, we have discussed that every person expresses 6 type of class 1 on his cells, nucleated cells and platelets, right? And these 6 type of molecules which are expressed on any person's any cell will be identical within the same person. Let me explain. If you take one of my skin cell, take one of my liver cell, take one of my neuron and take one of my nephron cell, on all of my cells the class 1 molecules will be of the same specificity. For example, if you take from Marla cells from any part of her body, all of them will have this specificity of class 1 molecule set. And if you take any cell from Ramirio, right, either from his liver or from his neuron, or you take a cell from his skin, all of them will have same set of class 1. Is that right? Now we come to, in this way what we have learned, that class 1 molecules act as molecular fingerprints on a person's cells. Is that right? Now we come to the class 2 molecules. Number 1, class 2 molecules are expressed only on antigen presenting cells. It's a very big difference. Class 1 molecules are presented on all the cells in the body and platelets, but class 2 molecule genes are only expressed in antigen presenting cells. Which are the antigen presenting cells? You know macrophages? Yes, macrophages. They are B lymphocytes. There are dendritic cells in spleen, for example, and Langerhans cells in the skin. So what are these cells? These are professional antigen processing and presenting cells. Is that right? Class 2 molecules, again, class 2 genes are present in all the cells, but class 2 genes are not expressed in all the cells. Look, class 1 genes are present in all the cells and they are also expressed in every cell. Class 2 genes are present in every cell, but they don't express in every cell. So it means that class 2 genes have selective expression. They are only activated in antigen presenting cells. Is that right? So it means all your cells will have class 1 molecule, but antigen presenting cells have class 2 molecules in addition to class 1 molecule. It means macrophages which are derived, if you bring some macrophages out of my body, they will have class 1 molecules plus they will have class 2 molecules. But if you bring, suppose one neuron out of my brain, if they are there, if you bring a neuron out of my brain, it will have the class 1 molecules, but it will not have class 2 molecules. Remember, every cell has every gene. But why different cells are different? Because different cells express different sets of genes. Is that right? While keeping that concept, class 1 genes are present in every cell and they are also expressed in every cell. Class 2 genes are present in every cell, but they are only expressed in antigen presenting cells. So only antigen presenting cells express on their surface class 2 molecule. Now, class 2 molecule genes, MHC complex for class 2 molecule also has 3 loci, 3 locus, that is D locus. Whole of this bunch of this genes is called D region. This was A region, this was C region, this was B region. A, C and B, these 3 regions together are concerned with synthesis of class 1 molecule. Then they found there is a D region. In the D region they have sub-regions. One region is DR, yes, DR, then there is D, Q and then D, P. It is not going in the normal alphabet fashion, right? So there is DR region and there is the DQ region and DP region. Now, actually these 3 regions will be on the maternal chromosome as well as they will be present on paternal chromosome. Now the point which you have to make it clear in your mind is, again, there are many alleles of DR region. As you know, A was not only A1, there were many A1, there were multiple alleles. Is that right? In the same way, DR genes are of many types, and every one on one chromosome number 6 has only one of those types. For example, Marla has DR1 here and she has DR4 here, right? She has DQ37 here and DP26 here, and let's suppose she has DQ7 here and DP, okay, she wants 12, I think someone told her 12 here. Is that right? Now we compare. So it means when Marla will express class 2 molecules, is that right? When she will express class 2 molecules, how many types of class 2 molecules she will express? 3 of paternal haplotype and 3 of maternal haplotypes, which will be only expressed on antigen presenting cells. So she will have DR1, she will have DQ, yes please, 37, and she will have DP 26. These 3 are from maternal haplotype, and from paternal side she has how many? DR4, DQ7 and DP12. So it means how many class 2 molecules she has? 3 maternal molecules, 3 from paternal, so 6 total. Is that right? But if we say how many class 1 and class 2 molecules Marla has on her tissues, so what you will say? 12. That is the point to understand. So every human being has how many type of MHC molecules on his tissues? 12. Is that right? Out of those 12, 6 are class 1 molecules and 6 are class 2 molecules. And 6 are maternal, 3 class 1 are maternal, 3 class 2 are maternal, 3 class 1 are paternal, 3 class 2 are paternal. Am I clear? No problem? Yes, you have a question.
In the case of the cells?
Antigen presenting cells. But every human being has antigen presenting cells. Of course, we will repeatedly say that class 1 molecules are present on every cell, but class 2 molecules are, in addition to class 1, expressed only on antigen presenting cells. So for example, if you remove my kidney, For example, you bring my kidney out. How many class 1 molecule and class 2 molecule it will have, my kidney?
12.
6 or 12? It will be. Do you think my kidney has macrophages?
Yeah.
Macrophages are the normal cells of the connective tissue of the body. Right? So my kidney, nephron cells mostly will have 6 class 1, but on macrophages of the kidney or mesangial cells of the kidney, right, you will have class 1 as well as class 2. So kidney tissue. If you take my kidney and put in the body of Mr. Lewis, how many MHC class 1 and class 2 molecules of my grafted tissue are going to Lewis' body? 12. This is very important. The problem is that Lewis' immune system will think these are some foreign proteins and he will react against that. Again, listen, let me tell you. But I will talk about that later. Let's complete class 2 molecule system on Ramiro also. Ramiro. There is DR, DQ and DP. Maternal and paternal side. DR, DQ, DP. Now, let's suppose on maternal side he has DR4 and for paternal side he has DR5, DQ, yes, 13, 33, DP on paternal is 21 and on maternal DP is, let's suppose, 18. Now you understand that there are so many alleles at DR region and every chromosome has one of those alleles. Is that right? Now, if I express class 1 and class 2 molecules, only class 2 molecules from maternal side expressed, you will tell me. And these are the paternal. Yes, DR, DR4, DQ, 13, DP, 18. Again, this is now paternal molecules, DR5, DQ, 33 and DP, 21. Look at all this. All the class 1 of Ramiro are different than the class 1 of the Marla. All the class 2 molecules on APCs of Ramiro, especially what are these, paternal, are different than paternal class 2 molecules on Marla. But just by sheer chance, let's compare this. What is this? Class 2 molecules from Ramiro's which paternal side or maternal side? Which one? Maternal side, right? And these are Marla's maternal side class 2 molecules, maternal haplotype. You see, just by chance, there is something shared in all this. Let's concentrate on this point. This is point of our interest, DR4. And you see here is also DR4. What does it mean? By chance, one type of MHC molecule is shared on both persons. Is that right? What we see that paternal allele on DR region, she was having DR4. Marla has DR4. And the maternal allele on the Ramiro DR region is again DR4. So if they are going to have a baby and this DR4 goes from Ramiro and this DR goes from R4 goes from Marla, so it will become DR4 in the baby. Is that right? Maternal and paternal. But another important thing is, if you are doing tissue typing or tissue matching, when we say that we are doing tissue matching, what we are really doing? We are trying to study these molecules on donor and we are trying to study these molecules, let's suppose, on recipient, and we see how many of these molecules are matching. If you look for the tissue matching between Marla tissue and Ramiro tissue, there is a very little matching. At which region? DR4 and DR4. but as far as other molecules are concerned, there is a mismatch. Is that right? There is a mismatch. Is it clear? Now we come to why these class 1 and class 2 molecules are so important when we talk about tissue transplantation and tissue grafting lesson. Actually, we take example of Marla. Marla has these special type of class 1 and class 2 molecules, right? When Marla's immune system was developing in late fetal life and early infancy, then Marla's immune system recognized these class 1 and class 2 molecule as self molecules. Marla's immune system recognized all these molecules as self molecules. Is that right? So right now, even though Marla is adult, her immune system recognizes only those cells as self cells, which cells are decorated with this specific type of class 1 and class 2 molecules. Is that right? Marla's immune system has grew up with this set of MHC molecules. So in case of Marla's immune system, only these special type of MHC molecules are considered self molecules. Is that right? Now let us suppose we take a tissue from Irene and transplant to Marla. Okay, not Irene. We start again Ramiro. Let's suppose, attention please, we take a tissue, for example kidney. We take kidney tissue from Ramiro and transplant in the body of Marla. What will happen? Actually, Ramiro's grafted tissue, donated kidney, has class 1 molecules which are totally different from class 1 molecules of Marla, and most of the class 2 molecule except this Dr are different than the Marla. So there's a big mismatch. So when you will put Ramiro's kidney in the body of Marla immune system, now let me draw, this is Marla's immune headquarter, immune head office. Now in Marla's immune head office, only these molecules are considered as self. When the kidney is transplanted, this immune system will recognize transplanted tissues, MHC molecules except DR4, they will recognize all other MHC molecules as foreign molecules. And immune system of Marla will start reacting against the grafted tissue as it is doing a very legitimate fight against the bacteria and viral and other proteins. And tissue will be damaged, transplanted kidney will be damaged, and it will be rejected. This is the mismatch, right, in the host tissue and donor's tissue which has created the problem. What we will say that there was no significant histocompatibility between the host tissue and donor's tissue. Now you imagine that let's suppose that Marla has a twin sister, monozygotic twin sister. If we take kidney from her, monozygotic twin sister of the Marla, if she is there, and we transplant that kidney in the body of Marla, Marla's immune system will recognize the grafted tissues, class 1 and class 2 molecules as self molecule, and it will not react significantly. Am I clear? Now, the biggest problem which is produced to us due to the class 1 and class 2 molecular system is that different people have different specific sets of class 1 and class 2 molecules. And in a given person, the immune system recognizes only those class 1 and class 2 molecules as self molecules with which the immune system has grown. Due to this reason, in my body, let's suppose I'm Dr. Najib, actually I am, If in my body I don't have any monozygotic twin, my mother never told me about that. So you bring a kidney of any person on earth and try to put in my body, there are good chances that there will be significant mismatch. Is that right? And what my immune system will do? It will attack the grafted tissue, as my immune system will recognize the MHC class 1 and class 2 molecules on the grafted tissue as foreign proteins and recognize them as foreign cells and try to destroy and eliminate out of my body. So due to this fact that every person has a unique set of specific set of class 1 and class 2 molecules which generally differs from others, right? There is a big problem in transplantation. Right now the surgical expertise for transplantation is very advanced. There are many tissues which we can transplant easily as far as surgery is concerned. The real problem is that tissue from the donor and the recipient do not match perfectly. Then what we do? For example, if Marla has to receive, okay, you come over here. Let's suppose now Marla is here and again Ramiro is here, as it was in the beginning of the lecture. Now listen, right. Now what is the thing? That let's suppose, unfortunately, Marla is very sick. She's diagnosed as renal failure, right? And one day Ramiro hears from someone, Marla has this problem. He becomes very emotional. He has some good memories. So he decides to donate one kidney to Marla, right? Now tissue is not perfectly matching. You know, the molecular signatures of MHC molecules on Marla tissue is different and Ramiro's tissue is different, right? Marla's immune system recognizes only Marla's MHC molecules as self. Marla's immune system does not recognize any other MHC molecules as self. So when we take the tissue from Ramiro, we take a kidney from Ramiro and put in the body of Marla, what will happen? Marla's immune system will be suddenly surprised that there's a big tissue with a lot of class 1 and class 2 molecules which are very different from her own class 1 and class 2 molecules. So her immune system is designed to spare all those cells which are having her own class 1 and class 2 molecule, and unfortunately her immune system is very much ready to destroy any cells which are having non-self class 1 and class 2 MHC molecules. So Marla's immune system will be activated against the grafted kidney coming from Ramiro as if a very big bacteria or something very big foreign thing is there, and immune system will start the attack. As doctors, we don't want this. So what will we do? What should we do? We put her immune system to the sleep and put her kidney in a secret way there, right? And then keep on suppressing her immune system, because whenever it will wake up, it will attack the grafted tissue. So what we do, we give lot of immunosuppressive drugs for many years and sometimes for lifetime if there is no matching at all. So she will be given lot of immunosuppressive drugs so that her immune system remains weak and suppressed, and it does not bother that there is a kidney with foreign MHC1 and 2 molecules. And of course, immune system is put to, what is this, suppression. Of course, the good point is it does not bother about the grafted tissue. The bad thing is that it does not bother about even bacteria, virus, fungus and other infections. So, if you are giving her immunosuppression so that grafted tissue is not attacked by her immune system, the price you are paying is very high, because now she will be very vulnerable to infections. Is that right? That is why, if time permit, we try to do the tissue matching. During the tissue matching, what we do, that we study the class 1 and class 2 molecules, especially in class 1 molecule we make special tests to determine class 1A and B molecules. And in the D region, especially we try to match the class 2 DR region, because these are very much immunogenic. So what we do, that when we are doing the tissue matching, we will take Marla's cells. I will tell you later in the lecture how we make the studies. And we also take the Ramirez cells, and we try to do HLA studies or MHC studies or tissue matching studies. In that we see how much their tissues are matching. Let's suppose if Marla she is very popular and good person, and she has 23 donors. 23 people are there willing to give her the kidneys. Then what we will do? We'll study what are the types of Marlas, MHC molecules, and then every donor will study what type of MHC molecules they have. And it is quite possible we come across some of the donor, or at least one donor we may come across, who has some of the MHC molecules matching with Marla. That person's kidney will be a better choice because there is some histocompatibility. Is that right? Because if MHC molecules are more matching between the recipient and donor, we say there's more histocompatibility. If MHC molecules have more mismatch between the recipient and the donor, we say there is histoincompatibility. And histocompatibility is mainly determined by MHC molecules. Histocompatibility is mainly, if you take her tissue and his tissue and try to match, then histocompatibility is mainly determined by MHC class 1 and class 2 molecules. That is why these molecules are called major histocompatibility molecules. You understand why these are called major histocompatibility molecules? It means there must be some minor histocompatibility problems also. Answer is yes. Now this is a very important concept, that if there are major histocompatibility, class 1 and class 2 molecules are matched, sometimes still there is little problem. And Marla's immune system still may attack the grafted kidney. Again, listen carefully. Sometimes what happens, then you may come across a donor by chance, by sheer good luck, that the donor has the same MHC class 1 and class 2 specificities which is in the recipient. And we are very happy we got a perfectly matched kidney. But that person is not genetically related with her, except that person is also a human being. So you take a kidney from that person and put into her body. Because there is perfect matching on MHC class 1 and class 2 level, so there will not be a great immune attack on the grafted tissue. But sometimes, even under these circumstances, after many years, grafted tissue may be destroyed and rejected. The doctors were surprised why it happened. Now they came to know that there are some minor histocompatibility proteins also. Actually, we should not focus only on class 1 and class 2 molecules. Grafted tissue has many other proteins also, other than MHC protein molecules. And other proteins may have 1 or 2 amino acid difference from Marla's protein. Yes?
Are these immune responses called type 2 hypersensitivity reactions?
We'll talk about that in detail later, because I will go into full detail what are the mechanisms, immunological mechanisms involved in transplant rejection. I'm just putting the foundation, right? We'll talk in detail about that. So what I'm talking about, sometimes even if you come across a very perfect matching as far as MHC class 1 and class 2 molecules are concerned, still sometimes in a very slow fashion over many years, rejection may occur sometimes. So doctors thought that there must be some minor histocompatibility proteins as well, for which we don't do the matching. And those proteins and their genes are not located on MHC locus on the short arm of chromosome number 6. Those genes and proteins, those genes are scattered on other chromosomes. Now they came to know those are just some other proteins in the body. In the grafted tissue, there are many other proteins other than the MHC1 and MHC2. And some of those proteins may have 1 or 2 amino acid differences with the similar proteins of recipient. So recipient immune system may recognize those minor differences in amino acid sequence in those proteins which are not MHC proteins, right? And if there are many mismatches, if there are many minor histocompatibility proteins coming in the grafted tissue, and many of them are different, all of them together may produce a strong challenge to Marla's immune system as foreign proteins. And then Marla immune system may gradually react against those minor proteins with minor differences, right? So because such minor proteins are also present, that is why these histocompatibility molecules class 1 and class 2 are called major histocompatibility molecules. Am I clear? Let's have a seat. Now we'll go into detail of how class 1 molecules work, and then we'll go in the detail how class 2 molecules biologically work in our body. Let's have a break.